We have to move forward now to our next speaker, Keren Klass. Excuse me. Keren is a first-year postdoctoral fellow at the Department of Ecology, Evolution, and Behavior at Hebrew University. And Keren will talk to us today about disappearing forests, genetic consequences for endangered monkeys.
Thank you. I'll just quickly share my screen. Okay, can everyone see that okay?
Yes.
Okay.
Yes.
Great. So thank you all for being here. Good morning. I'm just gonna jump right in. So we are currently in the midst of the 6th mass extinction. And what's different about this current extinction event is that it's directly caused by human activity, unlike the previous 5. So as you can see in this graph here, starting around the Industrial Revolution and more sharply from around the beginning of the 20th century, there's been a sharp increase in the extinction rates for vertebrate species. And this is compared to the background rate. So there's a natural rate of extinction that's always occurring just from natural processes. And you can see the dotted line, that's that background rate. So what we're seeing today is much, much higher than that. And this extinction crisis is being caused by many drivers. For example, pollution, habitat destruction, wildlife poaching and trade, and climate change. And the different effects of all of these can interact and exacerbate the threats that wildlife faces. So there are 504 recognized primate species in the world today. Primates are apes, monkeys, and lemurs. And about 60% of them are currently endangered, so at risk of extinction. And as many primates are arboreal, meaning they live in trees, the destruction and fragmentation of forests is one of the main threats to primates worldwide today. And forest loss and fragmentation can affect arboreal primates in many ways that can impact their chances for long-term survival. And this includes affecting their population demography, their feeding ecology, population genetics, predation risk, individual health and reproduction, and their ability to move in the landscape. So in order to effectively conserve this wonderful, beautiful diversity that we still do have in the world, we need to understand the specific ways that this main threat of forest loss and fragmentation is affecting different species. So we can then design and implement practical conservation actions that address the specific negative effects each species is experiencing. So my research topic, broadly, is how does habitat loss and fragmentation affect the movement and population genetics of endangered black howler monkeys? That's a lot of big terms, and I'm going to go in depth into all of them. First, starting with habitat loss and fragmentation. So up until quite recently, tropical rainforests, or just forests in general in the world, were these vast uninterrupted tracts of forest, and that this big green rectangle represents that sort of continuous uninterrupted forest. And animal populations, such as monkeys, were able to move through this forest freely. They lived in groups. Monkeys often live in groups that had their own home ranges where they could access resources. They can move around and access other areas if necessary. Individuals were able to move in between groups for new reproductive opportunities, and all this moving and mixing happened relatively unimpeded. With increasing human activity, these large undisturbed forests are becoming increasingly more fragmented into small and isolated forest patches. And these forest patches or fragments are separated by non-forest landscapes. For example, cattle pastures, different types of agriculture, towns and villages, roads. And this fragmenting and isolating of forest patches also creates a fragmentation of the animal populations that live in forests. So while monkeys might be able to move freely within their forest patch, if they do want to access resources or find new mates outside of the fragment they were born in, they often encounter serious difficulties in crossing these non-forest land cover types. And this isolation and fragmentation of populations and habitats can have many consequences for their ability to survive. So my study species is the black howler monkey. It's an endangered social, so group living arboreal primate, eats fruits and leaves. And what's really notable about howler monkeys as a group, because there are many species of howler monkeys, is that they howl. So I'm going to play a short clip, a sound clip of a single male howling. I will also note that they're considered the loudest land mammal in the world. So this is just one male howling. Were you able to hear that? Okay, so if you're standing under a tree and several males are all going at once, it can be quite deafening. So black collar monkeys, my study species, they're found in southeastern Mexico, in Guatemala and Belize. And previous studies have shown several negative effects of habitat loss and fragmentation on black collars. For example, increased predation risk. When they're forced to travel along the ground, they're often attacked by dogs when they have to move between fragments, as you can see in this picture here. And my own previous research has shown that for black collar monkeys in forest fragments, the population demography is actually quite different. And the main difference is that there are fewer adult males in forest fragments as compared to continuous forest. So where is my study site? I surveyed 34 forest fragments of varying size located around Palenque National Park. It's located in the upper inset map, the red dot, in Chiapas in Mexico. So I found 407 monkeys in these 34 fragments and collected all types of data while doing these surveys. And it's important to note that in this landscape, almost a quarter of forest cover has been lost just from the year 2000 onwards. So forest loss and fragmentation is a really serious ongoing threat in this location. And forest here is being replaced mainly by cattle pasture. So there's a lot of beef cattle raised in this area, as well as other types of agriculture like cornfields and palm oil plantations, and of course, roads and villages and other types of infrastructure. So how do I go about studying the population genetics of monkeys and forest fragments? So the first thing I did was collected lots of poop samples. So during my fieldwork from my PhD, from all the monkeys that I followed and surveyed and collected data, and I also collected these fecal samples, as you can see me doing here in this picture. I'll just mention that in the picture below, that's my husband, who also joined me during my fieldwork and was a great research assistant. Always found the monkeys first. So what's great about fecal samples is that they're non-invasive. That means that you can collect them without disturbing or harming the animal, as opposed to blood or hair, which you often have to capture the animal to get. With fecal samples, you just have to find what they leave on the ground, and it's much better for the animals. But the negative side of collecting DNA from fecal samples is that most of the DNA in fecal samples is actually bacterial because it passes through the gut. And so I worked together with a lab at the University of Calgary where they used a new method developed by Chow and Berge to extract and amplify the host DNA or the monkey DNA from these fecal samples as opposed to the bacterial DNA. So after extracting the DNA, it's being sequenced. Just a quick reminder, DNA is these double strands, long sequences of 4 bases, A, G, T, and C. And the sequences of these bases determines our genes, the proteins they produce, and the functions of our bodies. And the variation between these sequences, between individuals and populations, can tell us a lot about things like population diversity, divergence among different parts of the population, relatedness, inbreeding, and so on. So I'm taking all of this DNA that's been sequenced from the different individuals in my study site, and I'm applying bioinformatic analyses. So bioinformatics combines computer science, statistics, and biology to make sense of these really large and complex genomic data sets. And with these analyses, I'll be combining my results with demographic, habitat, and behavioral data to address 3 specific questions. So my first question is, how does forest loss and fragmentation affect gene flow and population genetics? So what are those things? Imagine an individual is moving from one forest fragment to another. They're literally taking their genes with them. And if they reproduce in that new location, their genes are seen to be flowing across the landscape from one location to another. But if that movement is restricted, over time, we'll see less gene flow, and we'll see that there begins to be this population genetic structure within the broader population. So within each circle, individuals are able to move and mix and reproduce. But between the circles, there's more genetic divergence. And over time, there will be less genetic diversity within each of them because there's less mixing overall. I also would like to understand how the genetic data can be informative about underlying changes to individual movements. So take this monkey up here in the corner, trying to decide, should I stay or should I go? If I stay, I might end up mating with close relatives, not great. If I go, this dog here might attack me on the way. But if I do manage to move successfully, I might end up here and I might reproduce. And then I come along and I sample that individual and other individuals from the fragment that originated from, and I see that they're related. And that gives me direct information about where monkeys are able to travel, if they're able to travel, what routes they take, and how far they can go. That together with the population structure data And when I correlate these types of information with the actual landscape itself, I can try to understand how specific landscape elements are shaping movement and gene flow. So I might discover that certain wide roads or villages are real barriers to movement for howler monkeys, whereas other types of land cover, such as cornfields, might not be so problematic for them. So then, if I have this type of practical information, I can then return to the local landowners whose fragments I surveyed and local authorities and give them some practical recommendations about how to improve the habitat on their land and improve the abilities of the monkeys to move safely through the landscape to maintain the gene flow and genetic integrity of the population, ultimately to hopefully improve their chances for long-term survival in this landscape. Now, I love monkeys and they're my thing. It's also really important to remember that these fragments contain countless other beautiful, important, interesting species. And so hopefully helping to maintain and conserve this habitat for the howler monkeys will also improve these other species' chances for long-term survival. So I just want to quickly thank my, my postdoc advisor, my PhD advisors, all my many research collaborators in different places in the world, the Azrieli Foundation, my other funding sources who have helped me along the way, especially the fragment owners who allow me access to their land, and my great research assistants. And thank you for listening. I'd be happy to take any questions.
Thank you, Keren. Very good. Have questions? Maybe. Is that a question or a clapping?
It was more a clap for now.
Okay. It was very interesting, Keren. And we have some questions. Ilan.
Yeah, thanks for that very interesting presentation. I had one question because I recently read about the idea of extinction debt, or the idea that the future extinction of a species has already occurred actually, and what we now have is some kind of debt that they're still walking on the earth, but essentially the story is over. Do you think that that's true for the monkeys specifically that you're researching? And does your research address this in any way? Thank you.
Yeah, so yeah, that's definitely something that I'm considering. So extinction debt mainly, what it means basically is that for long-lived species like primates, for example, we might only see the effects of threats that are affecting them now a few generations in the future. So it might seem like they're doing okay now, but in fact, they don't really have much chance to persist. So I have thought about that, and I brought it up in previous aspects of my research. Honestly, for this particular population, looking at the demography, aside from the fact that there's a weird lack of adult males, the population does seem to have a pretty good reproductive rate. And I found monkeys in almost every single fragment that I went to, which was surprising. There was a previous survey done in 2002, and they found monkeys in 50% of the fragments they went to. So they seem to be doing okay, at least for now. But since forest loss is ongoing and the fragments are getting progressively smaller and more isolated, it's very possible that if that doesn't change, then no matter what we do now, there just won't be enough habitat for them going forward. But it's inconclusive at the moment. It's like that's one of the problems with extinction debt. You only really, you gotta wait a few generations to see what the future will bring.
Thank you. Any other questions? Nadine, now.
Yes, we already talked so much during the workshop. That's why I feel like I already asked Keren everything that I want to know. But now I had another, I wonder, you focus like on monkeys mostly because you said like for personal reasons, but I get also practical and also probably theoretical reasons. I wonder how translatable like the results are to other species. You said in the end that you, of course, you want to translate like your research also to other species because you want to investigate in the long run the broader picture. And I wonder what kind of challenges they could be to do that, like on the long run.
Yeah, so actually, this type of research is done on many species, and that's one of the reasons that I wanted to sort of tackle this particular question and understand how to approach it and get experience with genetics and genomics, because this is really applicable to other species. I just like working with monkeys, so I'm doing that at the moment. But yeah, I think that any species that you can get genetic samples from, which is most, and some species are a lot more, are a lot easier to collect samples from, like insects, for example. They're easy to trap. Different animals, you can get samples of their tissue or their hair relatively easily. There are some studies that don't collect fecal samples at all. They collect hair samples, for example, left by different mammals in different parts of the forest. There are just a lot of different ways to approach it, and a lot of different methods are being developed to sort of deal with issues of small sample sizes or uneven sample distribution that you get from studying species in the wild. And this habitat loss and fragmentation problem is certainly not limited to primates. It's one of the main issues for wildlife worldwide. So it's really being studied in just about any ecosystem, any part of the world, all kinds of species. It's pretty, yeah, ubiquitous.
Hey, Keren, that was fascinating. I was curious to hear whether there are similar patterns in different habitats of the same species.
Yeah, that's actually, that's a good question, because truthfully, the same species in different locations can have different responses to similar threats. It really depends on so many different factors. It depends on the habitat quality, it depends on the configuration of the fragments in the landscape. It depends on what exactly the intervening landscape is made up of. It depends on the initial conditions of the population. How large was it? What was its demographic makeup? Are there any diseases that the population is dealing with? It's so complicated. So truthfully, while broadly, I think that my results can be applied to other Black collar populations elsewhere, just to see, for example, broad patterns. It's really hard to take specific results from specific populations and even apply them to other populations in different locations. Like I would have to know that the initial conditions were similar as well. They were dealing with other similar threats. The level of fragmentation was similar. It's what conservation is so tricky, because the same species experiences different things in different places. It's all very specific. It's part of the challenge.
Okay, are there any other questions? No, okay. And Keren, thank you very much.
Thank you.
Very interesting.